Welcome ladies and gentlemen to another video. I am Dylan Hamilton from C.A.R. Entertainment, and we are going to be continuing our revision of AS Biology tailored towards the CS spec. Today we're going to be moving on from lipids onto proteins, and then after proteins we're going to go to nucleic acids and then enzymes, and we will be done with the molecule section, and we'll be moving on to cells, which most people find the hardest about AS1 biology. So be patient and we will get there. We're going to be uploading one a day now for quite a while, so hopefully you will enjoy these videos. If one doesn't go up, it's because there's been a replacement video and it's way like an edit or something that we've brought out in the channel. So let's get straight into this. AS Biology, proteins. So we're going to start off by talking about protein structure and function. So basically the nature of proteins is that the proteins can function as enzymes, hormones and oxygen transporters. They form the bulk of the skin, hair, feathers, nails and cartilage. Proteins essentially are just huge 3D molecules whose building blocks or monomers are the variety of different amino acids found in nature. The three diagrams you see on the screen are amino acids, or proteins technically. So, protein structure and function. The proteins contain carbon, hydrogen, oxygen, nitrogen and usually sulfur. Sulfur is the distinction one. If you get asked a question about all the different types of macromolecules, you'll know which one's protein if it says there's sulfur involved. There's 20 different types of amino acids, and there's an infinite number of amino acid arrangements in protein. The function of most proteins is closely related to its shape, and shape of a protein is determined by the sequence of amino acids in the protein. The glycine R group is a single H atom, just for information, and alanine has a methyl CH3 group. This is just basic information given you probably won't be asked about it. We're now going to move on to a dipeptide. You can be asked a simple question about a dipeptide, and basically what a dipeptide is when amino acids are linked together by a peptide bond. This involves condensation reactions, which is the loss of water. The condensation reaction will take place between the amino group and the carboxyl group. The amino acids will join together forming a dipeptide. Now we're going to talk about all the different levels of protein structure, starting with primary and secondary. Basically, many amino acids will start by being joined together through peptide bonding, forming a polypeptide, and that's where we get the level of protein structure. The primary structure is the sequence of amino acids in the polypeptide chain linked together with peptide bonds. The difference though is the secondary structure has the O and the CO group being negative, and this enables the formation of hydrogen bonds. So there's an extra bond there, not just the peptide. And there's also two different types in alpha helixes and beta plated shades. In alpha helixes, the hydrogen bonds are formed between the amino acids occurring at regular intervals in the sequence. These bonds twist the chain of amino acids into a spiral shape, and these are held in an alpha helix shape by these hydrogen bonds, who help stabilize the shape. In beta plated shades, however, it's more rigid but less flexible, but it is oriented in an opposite direction, lying adjacent to each other. The hydrogen bonds formed between the CO and the NH groups. Now moving on to tertiary and quaternary. Tertiary is the main one. Basically, tertiary is the further folding of the secondary structure with a unique 3D globular shape, and a range of bonds are formed between the R groups. Myoglobin as an example. The four different bonds you'll need to know will be hydrogen bonds, which are relatively weak bonds, ionic bonds, which are formed between amino acids and the carboxyl groups. Some R groups are stronger than the hydrogen bonds, but are damaged by the changes in pH. Disulfide bonds, which are the strongest bond, they are covalent, and they are found between the R groups of the sulfur-containing amino acids. They're very strong bonds, and they are very important in giving strength. Other are hydrophobic interactions, which are usually located in around the R groups. Globular proteins also display a tertiary structure, and myoglobin is a globular protein found in muscle cells. In AS2 we'll talk more about myoglobin. The quaternary structure, it consists of two or more polypeptides of basically a tertiary structure, more or less. A conjugated protein, such as the glycoprotein or hemoglobin, is important in membrane structure, especially hemoglobin, as hemoglobin consists of four polypeptide chains, with each chain being attached to an iron-rich heme group, which is an essential part in the transport of oxygen. Once again, we will talk about this in AS2, and it is tied in with myoglobin very well. Now we're going to talk about protein groups. So fibrous protein groups are polypeptides arranged in chains to form fibers or sheets, with parallel chains linked by cross bridges forming a very strong stable molecule. These fibrous proteins are invariably structure and function. Collagen as an example. As collagen consists of three identical polypeptides that are held by hydrogen bonds, but they're found in tendons linking muscle to bones. Keratin is also an example of fibrous, which is in nails and hair. Globular proteins, however, are hydrophobic towards the center but hydrophilic towards the edges, meaning they're soluble in water. They have metabolic roles crucial. This is crucial in the role as an enzyme or antibodies, and hemoglobin is an example of a globular protein. Here's a comparison of the proteins if you get asked maybe in an essay or an example to compare to such. So basically basic shape between hemoglobin and collagen, obviously separate. collagen's insoluble, hemoglobin soluble, and so on and so on and so on. You can pause the video here if you want to go through the rest of the table. Now I'm going to talk about prions. Prions is a different type of protein, and basically prion is derived from protein and infection. Malformed proteins are called prions, and prions cause diseases such as TSC. BSE in cattle, CJD in humans, which means they lose physical and coordination. In humans, the memory and the body control are lost prior to death, so it is a fatal disease. It involves the replacement of a normal cell surface. The secondary structure of PRPC is composed of alpha helixes, while PRPSC is rich in beta sheets. The presence of PRPSC causes the normal PRPC to alter its secondary structure. Prions are stable as they're resistant to extreme temperatures and radiation, but prion disease is rare and it is unfortunately fatal. Prion disease arises in 3 different ways, and you could very well be asking this. First one is transmission, and this is for the consumption of infected food or an open wound exposed to the environment, with medical procedure involving infected material. Inheritance is another method, and this is a gene mutation occurring during meiosis, and it's a mutant gene coding for the synthesis of the mishapen prion protein. And finally, sporadically, which is the normal protein PRPC spontaneously transforming into the disease form PRPSC, with no evidence of inheritance or transmission, and the average age of this is about 65. Thank you for watching this video. Hopefully proteins has been a lot more broken down and easier for you to understand now. I've been Don from C.A.R. Entertainment, and I'll see you for the next video.